Thank you, Maya. Hello, my name is Krish Kumar, and I'm Kanishka Kumar. And this summer, thanks to HuBMAP, we were interns under the Bendel Lab and Angela Lab at Stanford University School of Medicine, which is the tissue map center of bone marrow. We're also undergraduates at Cal State Fullerton with a major in biochemistry and a minor in cellular and molecular biology. Today, we're going to be talking about the spatial proteomics of bone marrow tissue. So a little background about what we'll be going over today. So we'll be going over the background of bone marrow, the goal of the internship, the overview of MABITOF, and then we'll go diving down into the aspects of our experiments that we did, including antibody screening, QC gel, antibody labeling, OA technology, and MABITOF. So a little background about bone marrow. So bone marrow serves as the primary location of hematopoiesis, which is the production of blood cells, including white blood cells, red blood cells, and platelets. Within the bone marrow, there exist niches which play a role in supporting the transformation of hematopoietic stem cells, multipotent progenitors, and lineage-committed progenitors. The bone marrow can also be found inside the cancellous bone, where it contains a lot of red bone marrow, where rapid amounts of hematopoietic stem cells are found. So the goal of our internship was to learn the workflow of MABITOF through development of wet lab skills. So these wet lab skills could include preparation of buffers, antibody selection for MABI, antibody staining, and MABI staining. The scientific goals that we were focusing on were the titration of our new clones of anti-CD38 and anti-CD117 antibodies using metal conjugation and improving two markers that have been proven difficult in bone marrow / MABI using an anti-hapten antibody technology called OAO. So the methods we conducted, first off, started with immunohistochemistry, which is we wanted to detect whether our antibodies were present on the tissue. Then we went on to QC Gel / SGS page, which was seeing whether contaminants or BSA were present in our antibody. Then we went on to Antibody Conjugation / OAO, which is like labeling the antibody with a metal tag. And then we finalized the procedure with MABITOF, which is detecting multiple targets in a sample to image. So in terms of the samples that we collected, the Department of Pathology here at Stanford received about 1,800 to 2,000 biopsies each year, where roughly 4% of them are roughly normal. We specifically looked at sample of tissues located in the iliac crest, which is a part of the pelvis, from patient autopsies that had cancers in the red bone marrow like leukemia, lymphoma, and multiple myeloma. And in terms of the gender and racial ethnic groups that were related to the autopsy donors, there's a 1-to-1 male to female ratio, and the racial ethnic groups consisted of white, African American, Asian, and Hispanic. So MABITOF, it stands for multiplex ion beam imaging by time of flight. It's one of the newer techniques that was created by the scientists here at our lab, which uses mass spectrometry to image a biological sample. It can visualize up to 40 different proteins simultaneously, and it does this by using metal tag antibodies, which will bind to specific cellular markers in the sample of tissue. This sample of tissue will then be ablated by an ion beam, which will ionize those metal tags based on their time of flight, which can be detected and quantified by mass spectrometry. Mass spectrometry is a useful technique that can identify unknown compounds and ionize molecules into positively charged ions. Why MABI is so important and essential is because it enables highly multiplexed and sensitive immunohistochemical analysis of complex tissues, and it can track up to 40 different biological markers at the same time. It allows us to understand the cellular interactions of a tumor microenvironment, especially in the field of cancer biology. And the image displayed was mapped by our mentor Patricia using the MABITOF instrument. So the antibodies that we used for our project were CD38 and CD117, primarily because these antibodies show high expressions in the hematopoietic stem cells for red bone marrow biopsies that were sampled in the iliac crest. CD117 is a receptor for stem cell factor that has high expression in germ cells, hematopoietic stem cells, and early hematopoietic progenitors. CD38 is a glycoprotein that is found on the surface of many immune cells like B lymphocytes, natural killer cells, neutrophils, and myeloid cells. So next is antibody screening, also known as immunohistochemistry, which is IHC. IHC is a laboratory method that uses antibodies to detect antigen markers in a sample of tissue. So it's mainly used for protein localization and cellular architecture, and to make sure antibodies are present/active before you use them in antibody conjugation. So the first step of IHC consists of permeabilization, which is opening up those membranes of the cells so you can allow the antibodies to bind to the epitopes of the antigen. That is why you're using an antigen retrieval solution and dipping your slides in it so it can open up those epitopes. The second step is quenching/blocking, where it is reducing the chance of any non-specific binding. So that's why you're putting in a blocking buffer. The third step is primary antibody incubation, which is you dilute your antibodies in antibody diluent in zero dilutions with a dilution factor of 2, making titers of 1 microgram per mil to 0.5 to 0.25 to 0.125. And this is when the primary antibody is associated with a certain animal species, for example, like a rabbit or a mouse, where the paratope would attach to the epitope region of the antigen. The secondary antibody incubation is when the anti-animal species, for example, let's say anti-rabbit, is linked to the enzyme HRP and would bind to the Fc region of your primary antibody. The last step is visualization, where the main step is staining your tissue slide with a DAB, where you want to clearly see whether your antibodies are bound to your antigens under a microscope. And IHC is, although a step to visualize technically, it isn't a step you need if your antibodies have already been tested for. So we did an IHC titration of our anti-CD117 antibody followed by an H and E state. And we're going to be looking at a bone marrow TMA. We're going to be specifically looking at the myelodysplastic syndrome, MDS, which is pre-leukemia. So we did titers of a 1 microgram and a 0.25 to see what the difference was in the presence of antibodies. You can see that for CD117 in the 1 microgram, it is expressed in blast cells, which is immature cells, and that is not in normal cells. So the blast cells are actually a positive control for CD117 in a patient who has MDS. All the other cells are negative cells and serve as a negative control. So you can see there is positive cells and negative cells on the 1 microgram. And when you compare it to the 0.25 titer, you can see that it is much stronger here on the 1 microgram, and there's not that many antibodies present here. So you'd want to use the 1 microgram antibody for you doing the titer. We also did an IHC titration of anti-CD38 followed by an H&E stain. We're going to be specifically looking at the normal bone marrow, 80% cellular. We also did a 1 microgram titer and a 0.25 titer. And you can see that these bright brown cells are plasma cells. And the dim cells outside that, the less browning, is macrophages or developing B cells. And then there's a third layer, which is the dead negative staining, which is the blue slash purple color. So when you compare the 1 microgram titer to the 0.25 titer, you can see that there's not that much of a difference because there's 3 layers here and 3 layers here, which is what you want. So you can use the 0.25 titer for anti-CD38 so you don't waste 4 times the amount of antibody of that titer. So after the process of IHC comes the process of Antibody QC Gel, also known as SDS-PAGE. This is a common technique that is used to separate proteins based on their molecular weight. And it's mainly a step used for antibody validation and to check whether or not your antibodies contain BSA. The first step of Antibody QC Gel is protein sample prep. This is when you use TSEP, a reducing agent, to break down those disulfide bonds between the heavy chains of the antibodies, and then an SDS running buffer, which will denature those proteins, masking all the charges with a negative charge, making everything linearized and uniform. Next step is going to be SDS-PAGE, where you're loading those gels onto the wells, and then applying an electric current where these negatively charged proteins will migrate towards a positive charge. And you can see that the larger proteins will travel at a slower pace, whereas the smaller proteins will travel at a faster pace. After that is going to be gel fixation, where you pour a solution onto the gel, which will ultimately be used to sharpen and focus those bands on the gel so that they won't get washed away. After that is going to be gel staining, which is where you're pouring this, where you're staining your gel so you can visualize the proteins under the UV light. So this is an example of a QC gel that we ran using antibodies PARP, Vimentin, and TIGIT. And this was mainly ran to see whether these antibodies had BSA or not. And as you can see in lanes 3, 4, and 5, which contain PARP, Vimentin, and TIGIT, they do not contain BSA fragments at 50 kilodaltons. So it was good to go for our next step, which was antibody labeling, also known as metal antibody conjugation. This is an important and crucial step that many immunoassays incorporate, which is when an antibody directly cross-links with a label like a fluorescent dye, a fluorophore, or in this case, a metal isotope. The first step to metal antibody conjugation is going to be antibody buffer exchange, then it's going to be antibody reduction, and then it's going to be antibody wash, and then conjugation of metal and antibody, which is linking that antibody with the metal tag, and then final washing. So what you end up with this whole process is going to be an antibody labeled metal tag. However, when using antibody conjugation, conjugation has the potential to alter the antibody structure or disrupt its functions, meaning that its antibody antigen interactions could be affected, mainly because a lot of these commercial conjugation kits do not provide site-specific results. So this can result in a heterogeneous mixture, including many antibody conjugates, including aggregates, which is antibodies binding to other antibodies, non-specific binding, which is antibodies binding to other antigen markers, and non-functional antibodies. After antibody conjugation is going to be your lyophilization step, which is a freeze-drying step. This is a very convenient and safe method that is good for preservation, and it's good for long-term storage of your lab samples. Lyophilization, essentially, it dries your antibodies at very low temperatures in order to remove any damage from the products and maintain molecular integrity of the antibodies. So there's an alternate method to conjugating the antibody, which is a new technology called oil. Oil conjugation labels the antibody with an oil link, which is attaching to the primary antibody, and we add in a metal conjugated secondary antibody so it can be recognized by the MeBTOF. So how oil link works is that another name for it is protein G, and it is going to non-covalently bind to the Fc region of the antibody. So along with the protein G is a BPA, which is attached to it, and it's an N-natural amino acid, where the UV is actually going to catalyze the reaction of the BPA to the Fc region of your antibody. You can see here, there is the primary antibody here, and the oil link is actually attached to the primary antibody, where the hapten is going, and the hapten is along the oil, and the hapten is going to recognize the antihapten, which is on the secondary antibody, metal conjugated secondary antibody. And you can see that there's metal tags on that secondary antibody. So oil conjugation will actually result in 2 labels per antibody, 1 label per heavy chain. So this will result in 2 times the amount of metal antibody conjugation. So the steps for oil are, the first step is you're mixing your oil link regions with your antibody of choice. It's going to uncovalently bind to it. Your second step is you're going to illuminate your solution under a non-damaging 365 nanometer UV light, which is the optimal cross-linking time, and it's going to take 2 hours. It's also known as the LASIK method, the light-activated site-specific conjugation. And it's going to allow you to covalently bind, it's going to allow the oil link regions to covalently bind to your antibody. The third step is not necessary, but it is a bead purification step, which involves the use of water and metal beads. So the metal beads have an Fc region of antibody on them, and what it does is removes any excess oil in the solution after the UV cross-linking step. So one unique thing about Oyo is that it does not require the antibodies to be BSA free. So because the Oyo conjugation is site specific, that the disulfide bonds will not break in BSA, thus not making the protein G bind to it, so it binds to the specific antibody of interest. So BSA basically makes no difference. But a reason why you should add BSA to the antibody solution is because when you do bead purification, we lose a significant amount of antibody, and one way to avoid the loss of sample is adding a carrier protein like BSA. When you add a carrier protein like BSA, it avoids any loss of sample if your antibody has a low concentration. So when we were testing our antibodies, we were going to do it with Oyo conjugation, but we wanted to nanodrop them before that. A nanodrop is an instrument that determines your antibody concentration. So our values of stock solution, the concentration of CD38 before, it was 200 micrograms, and CD117, it was 128. When we nanodropped them, it was about roughly 200 micrograms per mil. When it does contain, and it did say it contained BSA, so we were a bit confused because when it usually does contain BSA, it is a 10 to 100 times concentration increase. So to make sure it did contain BSA, we did a QC gel. So we had a ladder as our reference, and we had a lanes 5 and 6 as our antibodies without BSA. We had a BSA control as a positive control. And you can see in lanes 3 and 4, those were antibodies supposedly containing BSA, and it did in fact contain BSA. You can see the 50 kilodaltons is the BSA line. And so why we did this QC gel was because we wanted to see why our nanodrop values was not accounting for the BSA in the solution, because normally our antibodies come with BSA, but the values that we got did not confirm that the nanodrop was not accounting for the BSA. And so now it was time to do the Oyo conjugation, where we picked CD38 to be conjugated to the AU1 hapten tag and CD117 to be conjugated to the flag hapten tag. After we conjugate them with Oyo and we illuminated them under UV light, we split the volume of the antibodies into one being purified with the metal beads and one being non-purified, just so we can see whether having excess Oyo would increase background noise on the MeBTOF instrument. We then nanodrop the antibodies. However, what we didn't realize was that because we had such low volume of CD38, there was a significant drop in concentration. As you can see, it went from 0.16 milligrams per milliliter non-bead purified to 0.01 milligrams per milliliter bead purified. Before doing this, we should have added more BSA to act as a carrier protein so it can be filtered out during the purification step. So we would have avoided a lot of sample loss. So then we performed a second QC gel to see whether our Oyo conjugations did work successfully on the antibodies, where we split the gel into two sides consisting of non-reducing conditions and reducing conditions. Reducing conditions meant that we would add TSEP, which would break down the disulfide bonds between the heavy chains of the antibodies, and you would see the separate heavy chains and the light chains. Non-reducing conditions was mainly to see what the overall conjugation state of the antibodies was, where we didn't add TSEP. An Oyo conjugated antibody would look like this, where a heavy chain is 50 kilodaltons, a light chain is 25 kilodaltons, Oyo is 8 kilodaltons, and the hapten tag is roughly around 1 kilodalton. So here are the results of the second QC gel that we ran, where lanes 2 to 7 are going to be your reducing conditions, and lanes 8 to 13 are going to be your non-reducing conditions. You can see here are going to be your heavy chains of the antibodies at around 50 kilodaltons. Your light chains are going to be around like 20 to 25 kilodaltons. And these bands over here are going to be your heavy chain conjugated to an Oyo tag. Lanes 2 and 3 contain the AU1 tag, and lanes 5 and 6 contain the flag tag. Makes sense because flag is heavier than AU1. So it makes sense that flag is higher up in the gel than AU1. For the non-reducing conditions, it was mainly to see what the overall conjugation state of the antibody was, and these are your non-separated conjugated antibodies. However, it's very hard to see that as these Oyo hapten tags are roughly very small, only about 10 to 12 kilodaltons. So when looking at MeBe, it involves the use of metal conjugated antibodies, which are labeled with a unique metal isotope. and it binds to a certain protein or cellular marker. And so these antibodies actually serve as a bridge connecting these unique metal isotopes to the targets of interest in a sample of tissue using mass spectrometry. So maybe imaging actually consists of 3 steps. The first step is antibody validation, which is maybe staining. So the tissue section is actually stained with a mixture of antibodies on the first day, each labeled with a unique metal isotope. You can see there is a lot of markers and antibodies that we mixed in. And then the second day is a second antibody mixture, including markers such as SMA, HH3, and DSANA, which are nuclear markers and attach to certain blood cells that so you can visualize the cell more. After that is going to be maybe image acquisition, where in order to detect these metal isotopes that are conjugated to these antibodies, the tissue is rasterized pixel by primary charged ions. And then the secondary ions that are released from the tissue are measured by mass spectrometry based on their time of flight to generate this n-dimensional image. So here are the maybe results. This is an optical image of the bone marrow TMA, where we look at TMA1 specifically, which is the healthy bone marrow tissue on the maybe slide that we use. In the maybe top, we were able to get this image where blue represents dsDNA, which is going to label the nuclei of the cells. The red here represents HLA1, which is a membrane marker, which is going to label the membranes of the cells. And then the yellow here is representing that CD38 signal, where you can see that it is surrounding the membranes of the cells. And this signifies that we did improve and get a better signal on CD38 through the maybe-TOF instrument. In this image, the red represents the iron. And one thing you can get without metal tags is a native biological element like iron, which does not need an antibody to be detected by maybe-TOF. So what do we find through this project that we did? So we improved our signal of CD38 in bone marrow tissue via the maybe-TOF, and we're still working on CD117. For further research, it's opening the doors to understanding the spatial organization of the hematopoietic niches in human bone marrow and map to cellular neighborhoods, which can be used to improve bone marrow transplantation and help us study leukemia and MDS. Maybe it's allowed us also to see the bigger picture of cellular landscape, while also drilling down to the details of individual cells, such as the areas in tumor microenvironments and areas around cancer. Maybe essentially contributes to a deeper understanding of biology and disease and findings of the cancers such as leukemia, lymphoma, and multiple myeloma. It's allowing us to map the spatial cellular composition and structure of bone marrow. So this internship experience at Stanford has brought me so much exposure and experience as to when learning about the different types of immunoassays, the many types of cancers in the red bone marrow, or even just how a wet lab runs. My favorite part in this internship was working with the pathologists, Dr. Ferda and Dr. Sai, to see what the antibody-antigen interactions were in the IHC slides and whether we were getting signal on the maybe slides. This internship has not only helped me finalize my goals of becoming a medical doctor in the future, but also pursuing an MD PhD where I can continue to hone my skills in a wet lab setting. I'm very thankful and grateful for this summer internship thanks to HuBMAP, and also excited to be applying for the extension where I can perfect my computational skills throughout the academic year. So my summer experience has been more than awesome because being part of a research lab has taught me so much about what a lab really is and hone my skills to work in a wet lab setting. Obviously, in the beginning, it was very hard for me to grasp the material, and I wasn't comfortable in handling the tissues when doing IHC. But when I did more hands-on stuff, I learned a lot by applying the material, which made me understand why I did it. Because of this internship, not only did this strengthen my pursuit of becoming a medical doctor, but also wanting to get an MD PhD. I feel like when I was doing the research, it made me question every single thing about why it works, and being curious inspired me to pursue a PhD. My future goals align with the research I like doing at Stanford, and with the help of HuBMAP, I can put my best work out there, which is why I'm going to be applying to extend my internship throughout the academic year. So we want to thank NIH, especially Maya, for really guiding us through the internship. We want to thank HuBMAP for accepting us and for this internship. We want to thank Stanford University School of Medicine for hosting us. We want to thank our PIs, Dr. Sean Bendall and Mike Angelo, for giving us this opportunity. We want to thank our amazing mentor, Patricia Favreau, for really guiding us through the project, really helping us through every step. And we want to thank the Bangelo Lab. Special shout out to Christine, Warren, Dr. Ferda, Mako, Angie, Dr. Sai, and Zumana for helping us during the experiments. Thank you. Thank you.